Have you eaten fermented food products today? If so, your taste buds have enjoyed the end result of a process called fermentation. Fermentation is the process by which anaerobic microorganisms break down pyruvic acid, a byproduct of glycolysis. There are two types of fermentation: alcoholic fermentation and lactic acid fermentation. Through fermentation, different microorganisms produce different end products. For example, yeast produce ethanol and carbon dioxide, while other microorganisms produce various acids and other organic compounds. All living cells go through glycolysis, the process by which glucose is converted to pyruvic acid. The purpose of glycolysis is to create NADH, an electron carrier, energy in the form of ATP, and pyruvic acid to be used in subsequent energy-generating pathways. After glycolysis, two molecules of pyruvic acid remain. In aerobic organisms, these molecules would be further broken down to generate energy in the Krebs cycle and later in the electron transport chain. But something different happens in anaerobic organisms. First, the two pyruvic acids are converted into acetaldehyde molecules and two carbon dioxide molecules. NADH then reduces the two molecules of acetaldehyde into ethanol. Drinking alcohol has been discovered multiple times by many different civilizations throughout history. These primordial brewers did not understand the exact process by which their alcohol was formed. They just knew that leaving fruits or grains inside sealed containers would produce a strange yet tasty, desirable beverage. Beer fermentation begins with the addition of wort, a slurry of dried, partially germinated grain called malt, and warm water into a vessel containing a mixture of yeast and water. Hops are also added to give the beer its flavor. Different types of beer have various specific environmental conditions at which they must be kept during the fermentation process in order to produce optimal taste and alcohol concentration. For example, ales need to be kept at 68 degrees Fahrenheit for approximately 2 weeks, while lagers need about 6 weeks at 48 degrees Fahrenheit. Other environmental factors include acidity and relative density. Once the ingredients have been added and the environmental conditions have been set, the beer is allowed to ferment for a predetermined amount of time, depending on the type of desired product. It is during this stage that the yeast begin the fermentation process. The yeast diffuse the glucose molecules into their cell bodies and begin to break down the sugar through glycolysis. After the ATP has been harvested, the yeast cells are able to divide and generate more yeast cells, thus allowing fermentation to continue. The fermentation vessel is connected to a long tube that allows excess CO2 to escape, but once the beer has reached a certain specific gravity, the tube is sealed to prevent the release of CO2 and pressurize the container. This is where most of the carbonation of the beer will come from. Once the fermentation is complete, the beer is cooled to allow all the yeast and other solids to settle at the bottom for filtering. After filtration, the beer is ready to be bottled and capped. Once fermentation is complete, the beer is cooled, and after that, it is bottled. During the bottling process, the beer must be kept away from oxygen as much as possible to extend shelf life and prevent contact with other microbes. The bottle is filled with CO2 before filling it with beer in order to pressurize the bottle and maintain carbonation. Next, the bottle travels to the capping machine where a jet of water shoots into the bottle before capping it in order to remove any amount of oxygen that may have entered the bottle. Now, the bottle of beer is labeled and ready to enjoy. Like beer brewing, winemaking also involves fermentation of glucose molecules into ethanol and CO2. The only difference is the source of glucose. In beer, the source is grain. In wine, the source is grapes, normally red grapes or white grapes. The first step in winemaking process is to harvest and crush the grapes into a mixture of juice and pulp called must. At this point, must that will become red wine is immediately filled into the airtight fermentation tanks, while must that will become white wine is filtered to remove the skins before entering the container. Next, yeast, sugar, and water are added to the fermentation vessel, and the mixture is cooled to approximately 40 degrees Fahrenheit to begin fermentation. Once the fermentation process is complete, the wine is filtered and stored in either stainless steel or oak barrels to age. After the aging process, the wine is filled into bottles, and the cork is placed in the bottle by creating a vacuum within the bottle that sucks the cork into the neck of the bottle. The bottle is then foiled and labeled. Very interesting concoctions can be made with the aid of fermentation. This process really highlights the relevance of microbiology in industry, entertainment, and everyday life.